Okay, so let's talk about electron transport chain. First of all, I would just like to say, maybe this is a good thing to know, but NAD plus is a cofactor and it can accept hydrogens and it can get reduced to NADH plus H plus. Similarly, we have FAD that can accept hydrogens and gets reduced to FADH2. So whenever we say reduced, that means that it has gained hydrogens. Then we have something called reducing cofactors and electron carriers, which would be NAD plus and FAD, and these are important for the ATP production, and we're going to talk a lot about this today. So when we say reducing cofactors, electron carriers, blah blah blah, then we're talking about this. Then we have hydrolysis of ATP gives you energy. And why does it do that? It's because it has these macroergic bonds. So storage of energy is in these bonds. Cleavage gives you the energy. So yeah. So as you can see here, we had these 3 phosphates, and just breaking the bond gives you energy. Because within these bonds, we have a lot of energy. Okay. So a little bit about ATP. It stores energy, we just talked about that, and it stores them through these macroergic bonds, and cleavage of them gives you energy. ATP is crucial for metabolism. Okay, so this is a good thing to know. Metabolism is anabolism and catabolism, and ATP will regulate them. So if we have high ATP, that will start catabolism, because then we want to break it down. And if we have a lot of ADP or little ATP, then that will start anabolism, because then we will build up ATP. And this is really important. So ATP can be produced in 2 ways. The first one is substrate level phosphorylation, which is the direct transfer of phosphate group to ADP, which happens in the mitochondria and in the cytosol. In the mitochondria, that happens in the citric acid cycle in step number 5, I think. And it can also happen in the cytosol, which is, as you know, it's glycolysis. Glycolysis will do substrate level phosphorylation. Second one is oxidative phosphorylation, which is the one that we're going to talk about today. So hopefully this will make more sense soon. But it happens in the intercellular membrane of the mitochondria. Yeah, and this one I just copy-pasted from the PowerPoint of our professor's lecture. But phosphate can be transferred from compounds with higher delta G to those with lower delta G only. Yeah. First of all, before we talk about oxidative phosphorylation, let's talk about substrate level phosphorylation. And substrate level phosphorylation, we can use 2 kinases for it. And kinase is just the transfer of a phosphate from ATP to a substrate, or vice versa. So when you see kinase, just remember it's the transfer of a phosphate. Yes, we have creatine kinase, which is an enzyme that moves the phosphate group from phosphocreatine to ADP, and we will get ATP. And this will happen in the cytosol and mitochondria, and this is the reaction. So phosphocreatine plus ADP, the phosphocreatine will donate one of its P's to ADP, and it will form ATP and creatine. And this is important for ATP homeostasis in muscle, but it is short term. The second enzyme that helps in substrate level phosphorylation is adenylate kinase, which moves phosphate groups from AMP ADP, so we can get ATP. So again, this is substrate level. This is direct. We have ADP here plus P, ATP. Like it's, and yeah. Well, actually, kind of the same thing happens but we'll get to it. Okay, before we talk about ETC, let's just talk about the mitochondria. So, okay. here we have the outer membrane of the mitochondria. Here we have the inner membrane, and here we have the matrix, and here we have the intermembrane space. Intermembrane space. Intermembrane space. Okay, so this outer membrane is not as permeable, or it is the outer membrane is more permeable to ions than the inner membrane. The inner membrane is highly impermeable to a lot of things, including hydrogen. And this is really, really good for the electron transport chain. And we will talk about that, but I just wanted to mention that. And the electron transport chain will be located on the inner membrane of the mitochondria. So here we have, and the electron transport chain will be made up of 4 complexes plus ATPase, which will be the 5th complex. But it's right here, it's on the inner membrane. So remember that. Okay, here I've written inner membrane has proteins that are important for ATP production, for oxidative phosphorylation. The amount, size, and location of mitochondria depends on energy needs of the cells. So if we have, for example, muscle cells, they will have more mitochondria than other cells that will not that don't need energy as much. Okay, so this little thing that I've written here, you can read for yourself, but okay, so oxidative phosphorylation, chemiosmotic theory. This is what it is basically. Here you have the mitochondria. Here, this layer right here is the inner membrane. Here we have the outer membrane. Here we have the matrix. Okay, so what happens is we have the main point of the electron transport chain is here we have the 4 complexes. There will be cofactors that will donate electrons to these cofactors, or to these complexes. So electrons will flow through the whole, like every single complex. And as electrons go through these 4 complexes, this will release energy. This energy that is released will carry hydrogens that are on the inside or are in the matrix. It will pump these hydrogens out to the to the inner membrane space, and this will cause for a concentration. It will pump them out, and this will cause for 2 things, both a concentration gradient of hydrogen ions, so we will have higher concentration of them here, and also a electrical gradient of hydrogens, because hydrogens are positively charged and they will be drawn to the negative inside of the membrane. So this will cause a electrochemical gradient that is really, really strong. Let's draw here the 5th complex, which is the ATPase. So because I told you guys that this inner membrane is highly impermeable, it will not let the hydrogens flow in or flow out. Therefore, the only way out is through these complexes, and the only way in is through the 5th complex. So this is really good, because this means that when these hydrogens will flow back in through the 5th complex, this will create energy for because to the 5th complex we have ADP, and then we will have inorganic phosphates that are floating around. When these hydrogens flow back in, the ADP and the ADP will be able to react with the P and form ATP. And so, oh, okay, here we have the fifth complex. Damn, okay, I didn't see that. Okay, so as you can see here, this is the ATP synthase. The fifth complex, hydrogens flow in, and ADP, which will create energy, and this energy is used to add ADP and form ATP. And this is what I've written here, so you can read that. Okay. Next thing. So the difference between substrate level and oxidative phosphorylation. So in electron transport chain, we will have oxidative phosphorylation. The energy needed to phosphorylate ADP is provided by the electrochemical gradient of hydrogens. When hydrogen flow back in through the ATP synthase, ADP plus Pi gives us ATP. Flow of protons down the electrical. So this is, if we would like to summarize oxidative phosphorylation, it will be that sentence. Then in glycolysis, we will have substrate level phosphorylation, which will happen in step number 7 and 10. And step number 5 in citric acid cycle will also be substrate level. This is supposed to say hi. So, okay, here I've tried. So, in step number 5, here is where we will produce GTP. And 1 GTP equals 1 ATP, so it's basically the same thing. Succinyl-CoA. So the breaking of succinyl-CoA bond will provide a lot of energy, and we have a GDP, and that will cause for the energy, or cause enough energy for us to bind an inorganic phosphate to a GTP, and we will form GTP. And so that will be called substrate level. So there's no electrons flowing in and out, left and right, okay? So it's basically just 1 plus 1. And that will be an example of substrate level. Okay, so proton gradient formation, why is it important and how do we get it? So it's important. Why have I written hue? Hi. Okay. Proton gradient formation. It's important because high concentration of hydrogens outside versus inside. When hydrogen rushes back in through the ATPase, this will give energy for... I'm sorry, I have been writing completely... Okay. ATPase, this will give enough energy for addition of inorganic phosphate to ADP, which is what we've talked about. But the proton gradient is important for that reason. And these, I'm actually a little bit unsure. These hydrogens that, yeah. Energy that is released by ETC complex is used to build a proton gradient across inner membrane. Yeah. But the main thing is, again, as electrons flow, they will release energy, and this energy is used, these 4 complexes used to pump hydrogens outside. Again, I'm just repeating myself. Each complex has electron carriers. Okay. Okay, so electron carriers can be FMN, FAD, which will accept 2 electrons and donates 1 at a time. So in complex number 1, for example, we will have FMN. And FMN will accept 2 electrons, and then it will donate it to the next electron carrier. An electron carrier, you can imagine like you're standing in a queue somewhere, I don't know, or like somebody has a ball, and this person passes the ball to the next person, and the next person passes the ball to the next person, etc. These electron carriers work as these people that are carrying the balls. So, so you have an electron carrier, and it's carrying an electron, and then it gives it to the next electron carrier, and that next electron carrier will give it to the next one, etc. So you will have FMN, FAD, for example. That is an example of an electron carrier, and it will accept 2. And but the rest, so we have cytochrome that will only carry one, iron sulfur clusters that will only carry one, and ubiquinone which will carry two, and we will talk about ubiquinone. But as the electron transport chain, so we will go here from complex 1 to complex 4. Complex 1 will have a higher ability of accepting electrons, because it will have these electron carriers that will accept more electrons. So here you will, so for example, complex 1, you will have FMN electron carriers. And that is called the reduction potential. So reduction potential will be high from complex 1, and it will be less as you go to complex 4. Yeah. Okay, now let's talk about each complex. I will not go into a lot of detail exactly what they do. I've written what they do here, so you can just read that. But I will just explain kind of what happens. So, okay. First and foremost, you have NADH plus H. This cofactor will come from, I don't know, citric acid cycle or glycolysis or somewhere. It will come, these cofactors will come from somewhere, and they will donate their 2 electrons to FMN. And FMN will then give these electrons to iron sulfur clusters right here, and the iron sulfur clusters will continue giving them away until we come to ubiquinone. Okay, so one thing to mention is, as the electron is traveling, so as it is traveling, that will lead to release of energy. This release of energy will cause for this part of the complex to change its conformation and actually be able to transport hydrogens on to the outside, to the intermembrane space. So that's why, so just the fact that electrons are flowing from complex 1 to 4, they will release energy and hydrogen can be pumped out. Anyway, so as the 2 electrons are traveling, and then they meet ubiquinone. Ubiquinone will accept the 2 electrons, and as it does that, it will also pick up 2 hydrogens and become ubiquinol. And that's what happens there. Okay, then we can go to complex number 2. So the electron transport chain can either start from complex number 1, or it can start from complex number 2. Complex number 2 is directly attached to the citric acid cycle, together with the enzyme succinate dehydrogenase. So here we will have, so if the citric acid cycle is right here, succinate dehydrogenase would be right here. And what happens here is that we will have the molecule FAD, or the cofactor FAD. And as succinate is oxidized to fumarate, it will release 2 electrons. And these 2 electrons will be picked up by the FAD, and they will then travel through iron sulfur clusters, and ubiquinone will pick them up, and it will become ubiquinol, and ETC, etc. So yeah, that's what happens there, and then we will get an FADH, blah blah blah. But the electrons that are released from succinate oxidizing into fumarate, they will travel to ubiquinone, and ubiquinone will be reduced to ubiquinol. Then we will come to complex number 3. So ubiquinone, okay, by the way, ubiquinone is an electron carrier. It's an electron carrier, and it will be inside, it will act in the inner membrane space, or not here, but in between. Yeah, you get my point. The thing about ubiquinone is it will carry these 2 hydrogens, and it will donate it to cytochrome C, which is another cytochrome C is another electron carrier, but it will only carry one electron at a time. So ubiquinol will donate one electron to cytochrome C, and then cytochrome C will go to complex number 4. In complex number 4, we will have copper A ions and copper B ions. Copper A ions will be the ones that will receive the electrons from cytochrome C, and copper B ions will be the ones that will give these electrons to oxygen and produce water. So that's what happens there. And as it does that, 2 protons are pumped out. And that is basically the electron transport chain. Just remember again that it's the flow of electrons. And here I've kind of highlighted electron because you can hear it in the name. Electrons that are transported through the chain, and as they do that, they release energy, and this energy the complexes use so they can pump out hydrogen. Yeah. And this is kind of a summary of exactly what they do, and it's a bit more detailed than what I've talked about here and here. So you can read that. Okay, so the 3 mechanisms of creating electrochemical gradients of hydrogen for the chemiosmotic model. So firstly, actively transporting protons across the membrane. Complex 1 and complex 4 will do that. Chemically removing protons from the matrix. So the reduction of ubiquinone and reduction of oxygen, and then releasing protons into the intermembrane space, which is the oxidation of ubiquinone. The chemiosmotic model complexes in ETC set up for proton gradient that drives the formation of ATP by the energy that is stored due to the gradient. Again, just repeating myself. Okay, so we have come to the end. And here we will produce ATP. And how does ATPase work, and where does ADP and PI come from, or an inorganic phosphate come from? So this ATPase, which will be the 5th complex, is made up of 2 things. It's made up of F0 subunit and F1 subunit, and also this gamma subunit, and also, yeah, that's actually what's important. So the F0 subunit will be located in the intermembrane space, and here is where the hydrogens will flow in. Yeah, and as they do that, we will go to the F1 subunit, which is a hexamer. So it has 6 subunits or something, and it has 3 alpha beta dimers, so they can be in open, loose, and tight conformation. And if they are in open, then they have no ADP and no phosphate. If they're in loose, they have ADP and inorganic phosphate bound, but they don't have any ATP. Like the production of ATP hasn't occurred. And tight conformation is when ATP has been created, when it has been bound. So this is important to know. Flow of 3 hydrogens down the electrochemical gradient makes the F0 and gamma subunit rotate, and ADP and phosphate get the energy to undergo condensation reaction and form macroergic bonds. So they will form the ATP. And ADP, and so where does these ADP and inorganic phosphate come from? They will come from 2 transporters. So we will have adenine nucleotide translocase, which is an antiporter. So it will transfer ATP out of the mitochondria and ADP into the matrix. And then the inorganic phosphate or we will have phosphate translocase, which will import hydrogens and a molecule that I cannot pronounce, H2PO4, into the cell, so ACE importer. And that's how we will get the organic phosphates, so we can produce ATP. Okay, now let's talk about the electron transport chain ATP calculations. So if we have 4 hydrogens, that means that we will produce 1 ATP. And if we have 6 hydrogens, then we will produce 1.5 ATP. 1 NADH equals 2.5 ATP, and 1 FADH equals 1.5 ATP. Okay, so let's count. How many hydrogens have we pumped out? When I say 4 hydrogens, that means how many have been pumped out. Okay, so if we start, because I said that the electron transport chain can either start from complex number 1 or complex number 2. If the electron transport chain starts from complex number 1, it will be due to NADH donating its electrons. And if it does that, then 4 hydrogens will be pumped out. Then, in complex number 3, another 4 will be pumped out. And then in complex number 4, 2 hydrogens will be pumped out. That means that eventually all of them will go through the ATP synthase and produce ATP. And that means that we have 4 plus 2 plus 2 plus 2, that equals 10. So from 1 NADH right here, so from 1 NADH that has donated its electrons, 10 hydrogens have been pumped out. This equals 2.5 ATP. But if we start the electron transport chain from complex number 2, that means that we will pump out 2 plus 2 plus 2 hydrogens, that equals 6 hydrogens, that equals 1.5 ATP. So from complex 1 to 4, 10 hydrogens are pumped out, and that equals 2.5 ATP. And then from complex 2 to 4, 6 hydrogens pumped out, that equals 1.5 ATP. And also, so that means that I'm just repeating myself, but 1 FADH will give you 1.5, 1 NADH will give you 2.5, so 1 NADH will give you more. That's it. And now to my least favorite part, regulation, inhibitors, and activators. So activators of electron transport chain is increase of NADH and ADP, which is great because now we can both start the electron transport chain and make ATP. So if we have a lot of NADH, that means that we can start the chain. And if we have a lot of ATP, that means that we can actually make ATP. So that's good. Inhibitors. Okay, so we have an inhibitor called rotenone that will inhibit NADH from donating its electrons. So it blocks the transfer from NADH to ubiquinone in complex number 1. Then we have another inhibitor called antimycin, which will block electron transfer from ubiquinone to cytochrome C in complex 3. And then the third inhibitor we have is cyanide or carbon monoxide that will bind to the heme group A3 copper B, and oxygen cannot get reduced, which is, I think we've all heard of that. And then we have the IF1 protein that will bind to ATP synthase when hydrogen is in high concentration in the matrix. We want to conserve ATP. ATP synthase cannot hydrolyze ATP. If ATP synthase is inhibited, NADH accumulation inhibits PFK1 from glycolysis. Awesome if we want to conserve energy, so klart. Okay, this means of course. Yeah. Wait. Oh shit. So cyanide and carbon monoxide will block this subunit, or like this copper B, and copper B will not be able to reduce oxygen, and that's bad. Okay, so the relationship between electron transport chain and ATP synthesis. So electron transport chain can happen regardless if ATP synthesis will take place or not. But ATP synthesis cannot occur if electron transport chain is not happening. The thing about this is we can have something called uncouplers. So uncouplers, so as I told you once, that if we have a lot of hydrogens, they will only flow back into the intermembrane space through the ATP synthase. That was the only way for them to go down the electron. That was the only way for them to pass the inner membrane, because they were so highly. Because the inner membrane space is highly impermeable. But the thing is that we have something called uncouplers, and uncouplers will make another way for these hydrogens to flow back in. And if the hydrogens flow back in through another way than ATP synthase protein, then the hydrogen electrochemical gradient will dissipate, and we will not produce any ATP. So this can happen in babies or newborns when they have to create heat. So when we do that, we actually create heat. And uncoupling is observed in brown adipose tissue. So that is because there will be an elevated expression of uncoupling protein 1. So UPC1 is a protein, let's say for example, so this one, for example, will give an alternative way for hydrogens to flow in, and that will create heat. Let's talk oxidative stress. So let's just talk some basics. Oxidative stress is when you have an imbalance between reactive origin species, which is molecules that damage DNA, proteins, membranes, and antioxidants. And it is molecules that prevent ROS molecules from damaging. So ROS will damage, and antioxidants will prevent ROS. If we have more antioxidants, then, well, no, I don't really think in that context, but if we have oxidative stress, that means that there is an imbalance between the two of these. Okay, so that's one thing that's important to remember. A free radical is a molecule that has unpaired electrons. Now, what does this mean? So here you have a happy oxygen molecule. It has all of its electrons. It isn't very reactive because it's happy. But if we have a reactive oxygen species, that could be, for example, superoxide anion, which means that instead of just having, like, it will get an extra electron from somewhere, and that will make it so if this oxygen will get an extra electron right here, it will become highly reactive, because now that it has one extra electron, it will need a second one. So it will react with anything, and it will steal their electrons. And that is bad because if this superoxide anion is close to, let's say, a DNA molecule, it will come and it will react with the DNA molecule and steal the electron from the DNA molecule, and then you have a damaged DNA. So that's bad. There can be other reactive origin species. It could be, oh shit, it's oxygen species, not origin. Reactive oxygen species. So for example, peroxide or hydroxyl radical or hydroxyl anion. Yeah. Now, where do these reactive oxygen species come from? They will come from mostly from the electron transport chain, because 1 to 3% of them get leaked. Complex 1 releases superoxides to the matrix, and complex 3 releases ROS to the matrix and to the intermembrane space. Then we have ubiquinone that does partial reduction of non-complex 3 targets. So yeah, it will give away electrons to stuff that it shouldn't give electrons to. Then we have NADPH oxidases, which are important in phagocytosis, and phagocytes, membrane-bound enzymes that give an electron to oxygen destroy bacteria by production of ROS. And then we have ER, so byproducts of protein folding. So when we're doing protein folding in the ER, there can be some byproducts that will be ROS. And xanthine, I don't know how to pronounce that, oxidase helps in purine metabolism, but it can also cause ROS. So oxidative stress, ROS versus antioxidants. So some common ROS molecules is superoxide anion, which is this one, or hydroxyl anion, this one. We also have hydrogen peroxide, which can diffuse through the membrane and can be a signaling molecule. So a high concentration of hydrogen peroxide can actually cause, if there's a lot of it, apoptosis, but it can also cause for cell differentiation. I've written that really wrong, I think. Proliferation. Why is it harmful? Why are these ROS molecules harmful? Because they always want one more electron, and they will take these electrons from membrane or DNA or proteins. So the damage they can do. So if you have, for example, a hydroxyl anion, it can make damage to the membrane lipids and make malondialdehyde. Hydroxyl anion can react with bases, most commonly guanine of DNA, and with DNA reparation, this could lead to point mutations. Yeah. But thank God we have antioxidants. And antioxidants can be enzymatic, and they can also be non-enzymatic. So common enzymatic antioxidants are catalase and peroxidase. And that is really good because peroxidase or hydrogen peroxidase will neutralize hydrogen peroxide and will create oxygen and water, specifically water, thanks to the substrate glutathione on peroxidase. It donates the electrons and hydrogens to the ROS, which is in this case hydrogen peroxide. Non-enzymatic antioxidants is vitamins E, C, and K. Vitamins can be reoxidized by other antioxidants. So if we have a vitamin that has been an antioxidant and it has donated electrons, then another vitamin can come and donate its electrons to that vitamin, and that vitamin then can redo its functions. In proteins, amino acids can be oxidized, most common those with sulfur. Yeah. You can read this. Okay, then we have microelements and reducing cofactors. So this is important. Selenium is important for enzyme glutathione peroxidase or GSH peroxidase family. So we talked about hydrogen peroxide, and hydrogen peroxide can be neutralized by this GSH peroxidase family and glutathione peroxidase. And in this complex, we will have a microelement, which is selenium. Zinc is important for superoxide dismutase, so for SODs. Oh yeah, I haven't talked about that. But superoxide dismutase is an antioxidant that will neutralize superoxides. So oxygen, shit. Okay, this is too okay. And catalase will contain iron. So we said that enzymatic antioxidants is catalase and peroxidase, and catalase will have iron. Yeah, and that is it for me, and thank you so much. And if you have any questions, just write to me, or if I've said anything wrong in the video, just write to me and I will try to correct myself.